Welcome to This Explainer. Today, we're exploring a paradigm shift in human health that sounds almost like science fiction, but it is very real. We're looking at how the microscopic ecosystem living inside your gut might actually dictate how your body fights cancer. We're going to explore how our body's internal terrain interacts with disease and how natural compounds, probiotics, and repurposed drugs are actively being studied to modify this environment.
So, let's dive right in by asking a truly fascinating question posed by researchers. Could manipulating our gut bacteria actually improve how our bodies fight cancer? Current research is strongly suggesting that the microscopic life inside our digestive tract might just be holding the steering wheel when it comes to our immune response. Section 1. The Cancer Microbiome Connection. To really wrap our heads around this, we first need to look closely at the complex ecosystem living inside of us.
We're talking, of course, about the microbiome. For the longest time, the microbiome was thought of mostly as just an organ for digestion. But it isn't just that. It's actually a critical regulator of our entire immune system. The literature we're diving into today shows it's intimately linked to cancer risk, and it even dictates how well a patient might respond to cutting-edge treatments like immunotherapy.
And this quote brilliantly illustrates the concept. Naturopathic Dr. Nasha Winters describes our gut as an orchestra. Think about it. When the violinists and drummers are in perfect sync, your internal terrain stays healthy and tuned. But if the musicians start playing the wrong notes, well, the whole system descends into chaos.
Section 2. Repairing the gut barrier. What exactly happens when that biological orchestra starts playing completely out of tune?
It leads to a state called dysbiosis. Now, maybe you've experienced this yourself after a round of antibiotics, a poor diet, or even a period of high stress. That incredibly delicate bacterial balance gets thrown off, and when this happens, pathogenic microbes can multiply, break down our natural defenses, and actually start producing cytotoxic substances. These substances essentially fuel a cancer-promoting environment through chronic inflammation.
You can visualize our intestinal epithelial barrier as a sort of inner skin. It has these vital structures called tight junctions. They act just like molecular gaskets, sealing our intestinal cells together to strictly dictate what is allowed to pass into our bloodstream and what absolutely needs to stay out. In a state of dysbiosis, these critical gaskets can become severely compromised.
So the big question is, how do we keep those molecular gaskets tightly sealed? Well, our good bacteria ferment undigested dietary fiber and turn it into short-chain fatty acids. The rockstar here is one called butyrate. Butyrate is incredible. It acts as the main energy source for colon cells, possesses powerful anti-inflammatory effects, and it even works as an epigenetic regulator. That means it actually helps turn off the specific genes that allow cancer cells to multiply. Pretty amazing, right?
Section 3. Prebiotics and probiotics explained. How do we actively repair this gut barrier and restore the balance of our internal ecosystem using natural interventions?
The crucial point here is understanding the fundamental difference between probiotics and prebiotics. Think of probiotics as the actual live, beneficial microbes themselves. These are the seeds you plant in a garden, like Lactobacilli and bifidobacteria. Prebiotics, on the other hand, are the essential food or fertilizer, things like inulin or specific plant fibers, that help those seeds thrive, take root, and establish dominance over the bad bacteria.
Section 4. Nature's anti-cancer arsenal. Beyond just supplying beneficial bacteria, scientists are deeply researching how complex compounds found in medicinal plants and fungi can aggressively modify this internal terrain to make it highly inhospitable to cancer.
Take the mechanisms of botanicals like malaca and berberine, for instance. A single plant can contain dozens of synergistic anti-cancer chemicals that block the GLUT1 gene, which effectively starves tumors of the high glucose energy they desperately need to grow. But here is where it ties beautifully back to the microbiome, and specifically to a highly beneficial bacteria called Akkermansia muciniphila. What's absolutely fascinating about bioavailability here is that many of these plant chemicals actually have very poor absorption in our small intestine. But guess what? That isn't a flaw, it's a feature. It means these compounds purposely travel intact all the way down to the colon. Once there, Akkermansia literally feasts on them, multiplying rapidly and fermenting these botanicals into new, highly active anti-cancer chemicals. In fact, clinical studies are increasingly showing that a high abundance of Akkermansia muciniphila in the gut is a major predictor of whether a patient will respond well to cutting-edge cancer immunotherapies.
And it's not just plants. Studies on medicinal mushroom-derived compounds like AHCC show they act as powerful immunomodulators. They essentially enhance our immune surveillance by cranking up the activity of natural killer cells and T cells. These aren't just laboratory observations either. Real-world clinical trials have indicated that these compounds can lead to significantly prolonged survival in patients with certain types of advanced liver cancer.
Section 5. Repurposing existing drugs. Alongside nature's botanical arsenal, researchers are also investigating how to repurpose existing, well-tolerated prescription medications to target those exact same cancer pathways.
This brings us to drugs like ivermectin and mebendazole. Now, traditionally, these are known globally as standard antiparasitic medications. However, preclinical studies have demonstrated they possess a really potent ability to inhibit cancer cell proliferation. They disrupt the cellular microtubules of tumors, block major signaling pathways, and importantly, they selectively target cancer stem cells. Those are the critical cells responsible for tumor recurrence and resistance to therapy.
Section 6. Real-world clinical outcomes. But what happens when we take these repurposed drugs out of the petri dish and give them to actual human patients? To see how this translates, we can look at data from a recent prospective observational cohort study.
Let's break down the design of this study. Researchers followed 197 adult patients who were dealing with a variety of cancers like prostate, breast, and lung. They were prescribed a compounded combination of both ivermectin and mebendazole via a U.S. telemedicine platform. These patients took this combination alongside their standard therapies, and researchers closely tracked their outcomes over a quick 6-month follow-up period. The results? They were absolutely striking. At the 6-month mark, the researchers recorded an 84.4% clinical benefit ratio. That means in over 84% of patients who completed the follow-up, the disease either stabilized, shrank, or disappeared entirely. Breaking those self-reported outcomes down a bit more. Nearly 33% of those patients reported having no current evidence of disease. Another 36% reported that their disease remained completely stable, and over 15% saw active tumor regression. Furthermore, the medication had a highly favorable safety profile. The side effects were predominantly mild, meaning over 93% of the patients who experienced them were able to just adjust their dose slightly and smoothly continue their therapy.
As we analyze the future of these integrative therapies, from feeding our gut microbiome and utilizing plant compounds to repurposing incredibly safe medications, we have to ask ourselves a profound question. Could meticulously tending to your body's microbial ecosystem and internal terrain become the ultimate strategy for preventing and fighting chronic disease? It's a massive paradigm shift, and one that puts a tremendous amount of power directly back into your hands. Thanks for joining me on this explainer, and I hope this gives you a powerful new lens through which to view your own biology.